Hello, good evening, everyone. Has everyone joined? Let's wait for 2 minutes for other students to join, then we will start the class. Okay, just let me know, is my voice audible to everyone? Good evening, Deepa. Okay, thank you, Anand, and very good evening. Is the screen visible to everyone? Okay, thank you, Akshat. So, everyone, good evening once again, and welcome to the R programming course. So, first of all, thank you so much for joining this course, and I'm really glad to have all of you here. So, before we start learning R, I would like this to be an interactive class. Okay, I don't want this to feel like a one-way lecture. So, if you have any questions during the class, please don't hesitate to ask. So, there are no silly questions here. If one student has a doubt, chances are that several other students have the same doubt as well. So, before I introduce myself, I would like to know a little bit about all of you. So, let me know what's your name, what's your background, what's your education qualification, anything about you, whatever you would like to introduce yourself. For example, where are you from and why did you join this course? So, let me know a little bit about all of you. So, let me know about you. So, does anyone want to introduce himself or herself? Hello, Dr. Siddesh. So, everyone, we have Dr. Siddesh as well among us. So, he has a PhD and he's a very intelligent person. Thank you, Dr. Siddesh, for joining. Anand also have PhD. Okay, Anand is working as assistant professor. That's a very nice profession, Anand. Okay, so Dr. Siddesh and Dr. Anand both are virologists. That's nice to know that we have some talented people in our community. What about others? Okay, Aryan Shukla is doing BSc in Life Sciences. Okay, very good. Anand is wants to be biotechnologist. Okay, so do we have someone else from BSc or MSc or from any other background? Okay, Himani is pursuing MSc in Biotechnology. Okay, does anyone else wants to introduce himself or herself? Okay, from YouTube Live, we have Nagesh and he's doing MSc Microbiology. Okay, very nice, Nagesh. Okay, so thank you everyone, and it's really nice to know a little bit about each of you. So let me briefly introduce myself. So my name is Muskan Kashyap, and I am a postgraduate in Masters in Biotechnology, and I work in the field of bioinformatics. So I have worked on several bioinformatics projects which involve the biological data analysis, the variant analysis, genomics, and particularly programming. So I also run Biotechshalaa, where my goal is to make bioinformatics and computational biology easier for life science students. So one thing that you will notice throughout this entire program is that I like teaching concepts in a simple way. So I don't assume that you already know something about programming or any other concepts. So we will build everything step by step. And my goal here is not to teach you just R syntax, it's to help you understand that how R is actually used to solve the biological problems. So, before we begin, I would like to set a few expectations. First, this course starts from absolute basics. So even if you have never written a single line of code before, so that's perfectly okay. Second, we will not just learn theory. We will work with real biological data sets and we will perform actual data analysis. Third, I encourage everyone to practice after every class because programming, it is a skill. So you can't just learn it by watching the video. So the more you will type, the more confident you will become. Okay, are we clear with the third expectation? And the lastly, the fourth, if something is not clear, you can ask me your doubts without any hesitation. Okay, I would rather explain one concept three times than have someone leave the class confused. So are we clear with these expectations? So they are very simple, but you need to follow them. Okay. Okay, great. So now that we have gotten to know each other, let's begin with our lesson one. So before we understand what R is, let's first understand why we are learning it. So let's imagine a situation. Okay, we are imagining a situation and suppose that you get a job as a bioinformatician. Maybe you can get a job as a bioinformatics analyst or genomics analyst, any position. Okay, so just imagine that you get a job as a bioinformatician. Now, on your first day, your senior gives you a file, and that file contains gene expression data. Okay, your senior gives you a file, and your senior says that this file contains gene expression data from 30,000 genes. And now he gives you one task. And your task is to compare the healthy patient genes with cancer patient genes, and you need to tell which genes are different. Do we understand the task? First of all, let me know, do we understand the task? Okay, very good part. What about others? Okay, very good, Anand. So, our task is to compare the healthy patient genes with the cancer patient genes, and we need to tell that which genes are different. Now, first of all, what you will do, you will open that file, obviously. Now, that file looks something like this. Okay, here we have gene, here we have patient 1 who is healthy, here we have another patient who is healthy, here we have another patient who has cancer and another patient who has cancer. So this is the table of the gene expression data. Now, we are not talking about 10 genes or 100 genes, we are talking about 30,000 genes and maybe the data from the 200 patients. So you cannot compare all of this manually. Tell me, can we compare all of this manually? Because we are talking about 30,000 genes and your task is to compare the healthy patient genes with the cancer patient ones. And there are 30,000 genes and the data of 200 patients, which means manually we cannot compare this because it would take too much of time. Yes, Dr. Siddesh, it is not practical enough, right? So what do scientists do? They use computers. Okay, scientists, they use computers for these kind of tasks. But computer does not understand English. Computers, they understand instructions. Do we understand? When I'm saying computers, they understand instructions. For example, on the top right, you are seeing Biotechshalaa and I want to move this cursor to Biotechshalaa. So what am I doing? I will give instruction. I will use the mouse like this. So here, what are we doing? We are giving instructions. Each and every time we give instructions to the computer. Now, again, computer does not understand English. So how computer is understanding our instructions? So a programming language. Now we are talking about a language which is called a programming language. So a programming language is simply a way of giving instructions to a computer. And one of those programming languages is R. So what is R? It is a programming language. Now, what does R actually do? So you can think of R as your assistant. Okay, think of R as your assistant. So for example, instead of saying that I will calculate the average expression of all of these 30,000 genes, which is practically not possible because it will take too much of time. So instead of saying that I will do this work myself, you will tell R that please calculate the average of all of this data. Now, in the same way, instead of saying that I will make the graph myself using the pen and paper, you will tell R that please make the graph of this data. Okay, and again, R will do all of this work in seconds. So R here, it is not replacing your biological knowledge. Okay, it is a very important line and a very big misconception. R here does not replace your biological knowledge. It helps you apply it to large data sets. Do we understand it? Do we have any question, any kind of confusion? Okay, now why R has become one of the most widely used programming languages in biological research? Why is R so popular in bioinformatics? So let's think about what a bioinformatician does every day. So a bioinformatician, he or she works with biological data. Okay, in the simple line that you can describe the work of a bioinformatician is he or she works with biological data. Now, what is this biological data? So DNA sequences, RNA sequences, protein data, gene expression data, or clinical data or mutation data, all of this is the biological data. Now, these data sets, they are often very huge. Just like we saw the example here that we have the data of 200 patients and we have approx 30,000 genes, which means the data is pretty huge for us to do all of this work manually, right? So sometimes this data, it can contain millions of values. That means 30 million genes. Let's just assume that we have 30 million genes. So the data is very huge and humans, they cannot analyze that much information manually. So R here, it was built for analyzing the data. Do we understand why bioinformaticians use R? Because R here, it was built for analyzing the data, and that is why scientists, they use R. Okay, how does R differ from Python? So Python and R both are programming languages, but they both have their own importance. Okay, I will also explain it later, but for now, just listen that R here, it is built for analyzing the data. Okay, I will explain this point in brief. Now, one common thing again that people think that I will learn R, or maybe you will think this as well, that I will learn R in this course, but where will I actually apply it? I will just learn the concepts, how to use R, but where will I actually use it? So let's take a look at here. Here we have few of the examples where you can apply R. So example number one is RNA sequence analysis. So don't worry if you don't know what RNA sequence analysis is, what gene expression data is. Don't worry about these things. Just listen and try to understand. Okay. So the first example that where we apply R is RNA sequence analysis. So imagine that you have RNA sequencing data. Just like here we have gene expression data, in the same way we have RNA sequencing data. So just assume that we have RNA sequencing data from healthy and cancer samples. And obviously the samples, they came from the patients. Okay. Now, your task, you have the data, the RNA sequence data. Now, your task is to answer all of these questions. And what are these questions? That which genes are more active in cancer, which genes are less active, and which genes should we study further. So R here, it will help you first of all read the data, because as we are talking, the data is very huge. So you need to understand what is this data. So R here, it will read the data for you. It will clean the data for you. It will analyze the data for you. And you can also create graph using R, and you can also find the important genes. So this is the first example, RNA sequence analysis, where you can apply R. Now, second example, DNA sequence analysis. Now, suppose that you have a DNA sequence and you want to study that DNA sequence. Okay, so just assume that this is that DNA sequence which you want to study. Now, you want to know that how long is this sequence, how many A, T, G, and C bases does this sequence contain, or does this sequence contain a particular pattern? So again, R here, it can answer all of these questions very quickly. Okay. Now, third example, if you want to generate figures. So imagine that you have completed an analysis. Just imagine any of the analysis that you have completed it, and you want to create a graph. Okay, you want to create a graph for the paper. Now, instead of drawing the graph by hand, R can create publication quality figures that researchers include in their scientific papers. And the last example is research papers that we just talked about here. So, have any of you ever read any published papers? Any published bioinformatics papers? Anyone? Okay, Parth have read one bioinformatics published paper. So in those bioinformatics published paper, if you read it, you will often see some statements like this analysis was performed using R. These are the statements that they are very common in those research papers. So just like this analysis was performed using R. We have statements like this in those research papers. So this is because many popular bioinformatics tools and many popular packages, they are written in R. That is why scientists, they use R a lot. So by learning R, you are also learning that how many research analyses are actually carried out. So these are the some key areas that you may apply R. Okay. Now, one more thing, that bioinformaticians, they don't only use R. R is not the only one. So this is something I want you to know from the beginning, that most bioinformaticians, they use several tools. Now here, Dr. Siddesh, I will answer your question. So for example, R. Okay, here I just talked about that most bioinformaticians, they use several tools. So one of the tool is R that we are talking about. So we use R for data analysis. We use R for statistics. We use R for visualization. Just the example we saw here, that we can use R for visualization. Now, another tool is Python. Now, why we use Python? We use Python for automation. We use Python for scripting. We use Python for machine learning. We use Python for sequence processing. So both R and Python are programming languages, but they both have their own importance in different kind of work. Okay. Now, another tool is Linux. So Linux, we use Linux if we want to run bioinformatics software, or if we want to work with servers, that's where we use Linux. And sometimes we also use SQL. Okay. SQL is used when we want to manage large data sets. So sometimes that tool is also used. So the point is, R here, it is one important tool in your toolkit. Okay, just assume that you have a toolkit, and in your toolkit you have one tool which is R, and it is not the only one. So we can understand it with one more example. So for example, let's take example of a doctor. So a doctor, he or she, they use stethoscope, right? They use stethoscope, and they also use thermometer. They also use X-ray machine. They also use blood test. They also use MRI scans, right? So each tool has a different purpose. So similarly, R here, it is one of the most important tools for analyzing the biological data. And Python is another, and Linux is another, and SQL is another. So we have different types of tools. Okay, we are referring it to as tools because we use them to solve our biological problems. So a good bioinformatician, he or she knows when to use each tool. So do we understand it, the difference, everything? Do we have any question? Do we have any kind of question? Okay, what about others? Okay, very good, Aryan. So I don't want you to remember a definition like R is a programming language. No, I don't want you to remember that. I want you to remember this idea that R, it is a tool that helps a bioinformatician, or that helps you to ask questions about the biological data and get answers very quickly. So everything that we learn in this course will help us to do that. Okay. Yes, Parth, we can do that as well. We can also generate graph and images in Python as well. But here, as I explained, that we use different tools for different kind of things. Just like I explained the example of doctor, right? If you have a fever, what you will use? You will not use stethoscope for a fever, right? You will use a thermometer. So in the same way, we have different biological questions, and for that we use different tools. And we are referring R, Python, Linux, we are referring these as tools. Okay, so before we move forward to our next concept, I would like you to answer these three questions. You can answer these questions in your own language. So the first question is, why can't bioinformaticians analyze data manually? Second question, before today's lesson, what do you think R was, and has your understanding changed? And the third question, Why do you think R is useful for someone working with gene expression data? Let me know your answers. Just write in your own words. Okay? Okay, very good part. Data is complex and lengthy. Okay, very good, Aryan. Because data is huge, so it cannot be analyzed manually. Okay, well received, Dr. Siddesh. Okay, very good, Anand. Okay, thank you, Dr. Siddesh. What about others? Have your understanding cleared about R? Yes, very nice part. Okay, Sahan, so has your understanding changed? Did you learn more about R today? Okay. Does anyone from the live chat on YouTube want to answer? Okay, we have Rabia from Bangladesh. Yes, very nice, Akshat. Okay, so here we understood that what kind of data we are dealing with or a bioinformatician works with. And the data is pretty huge. And once you understand the data, so learning R will make much more sense. Okay, we will learn everything step by step. Okay, so here in this question, we are talking about gene expression data. And the question is that why do you think R is useful for someone working with gene expression data? So what is this gene expression data? So let's talk about this, that what is gene expression? So this gene expression, it is one of the most important concepts in bioinformatics. So my question to you, okay, listen to this question very carefully. Does every cell in your body have the same DNA? Does every cell in your body have the same DNA? Your body has brain cell, your body has skin cell, muscle cells, liver cells, we have different kind of blood cells. So do they all have same or do they all have different DNA? Let me know the answers. What do you think? Very good, Deba. Deba, can I call you Deba? Okay, very good part. Same DNA. Ranjita, same DNA. Okay, Ragini, Akshat, same DNA. Okay, so almost every cell in your body has the same DNA, right? All of your answers are correct. Now, another question. If every cell has the same DNA, then why does a brain cell look different from a skin cell? This is the question now. If every cell has the same DNA, then why does a brain cell looks different from a skin cell? Who will answer? Yes, very nice. Dr. Siddesh, what about others? Let me know your understanding, your thinking. Okay, I don't want you to copy others' answers. Okay, very good, Anand. Okay, very good part. We have a new answer. Fate is different at specific level. What about others? Let me know what is your understanding about this question. That if every cell has the same DNA, then why does a brain cell look different from a skin cell? Okay, from YouTube live chat, Atharyan, different expressions, very good. Okay, so, okay, we have two more answers. Okay, very good, Himani. Okay, very good, Deba. Your answer is very nice that in developmental stage, different tissue have different transcription factors. Okay, very good, Ranjita. Very nice part. Okay, very good, Aryan. Yes. So the answer is because different cells, they use different genes. So all of your answers are correct because they are all linked together. So the simplest answer is that because different cells, they use different genes. We can understand it with an example for those who needs a better understanding to this concept. Okay. So let's understand it with an example. So just imagine that DNA is a cookbook. Do we understand what is a cookbook which have many recipes to create more dishes, right? So imagine that DNA, it is a cookbook. Now, the cookbook DNA, it contains 20,000 recipes, okay? And those recipes are the genes. We are referring those recipes as genes. So imagine a DNA as a cookbook with 20,000 recipes and those recipes are the genes. So now we have three kind of chefs. We have three different chefs. Now the chef number one, he works in a bakery. So if he works in a bakery, he will only use the recipe for cake, for baking cookies, or for baking bread, right? So from that cookbook, the chef one will only use those recipes. Now we have chef two who works in an Indian restaurant. So if he works in an Indian restaurant, he will use that cookbook for recipes like biryani, like dal or paneer. And we have third chef and he works in a juice shop. So he will use the recipes for like mango juice, orange juice, milkshake. Now, did the cookbook change? The cookbook did not change, means the DNA did not change. It is the same cookbook, means it is the same DNA. The only difference is that which recipes each chef uses, right? This is the only difference, that those three chefs, they are using different recipes from the same cookbook. Now, your body works exactly like this. Every cell has the same DNA, means the same cookbook, and each cell, they uses only the genes it needs. So for example, a muscle cell uses the genes which is needed for muscle function. A brain cell uses the genes which is needed for brain function. A liver cell uses the genes which is needed for liver function. So the DNA, it stays the same. The genes being used are different. Now, what does gene expression mean? So gene expression means a gene is being used by the cell. So do we understand the simplest definition of gene expression? That it means a gene is being used by the cell. So gene expression, it tells us that how much a cell is using a particular gene. That's it. So let's take a real example to understand it. So for example, we have gene A and we have gene B. So suppose the first gene that we have, it is gene A. Now, in a healthy cell, gene A is used a little. But in the cancer cell, that same gene A is used a lot. So here we will say that gene A has higher expression in cancer. So do we understand this example? Let me know, do we understand this line that gene A has higher expression in cancer? In the same way, we have one more gene, gene B. Now, in the healthy cell, this gene B is used a lot, but in the cancer cell, it is barely used. So, we will say that gene B has lower expression in cancer. So, when scientists, they compare the healthy and cancer samples, they often ask questions like which genes are being used more or which genes are used less. So this is called gene expression, or we can say gene expression analysis. Okay, and it is one of the most common tasks in bioinformatics, and we analyze its data using R. So we are taking the example of gene expression to understand R. So first of all, tell me, are we clear with this? Till here, are we clear about gene expression? Okay, so here we understood about gene expression. Now, where does R come in the picture? So, let's connect everything this to R. So, imagine you have information for 30,000 genes. The same example that we saw earlier, that you have a file of 30,000 genes. Now, for each gene, you want to know, is it used more in cancer, or is it used less in cancer, or is it there a meaningful difference? So we cannot answer that by looking at a huge table of information. So let's open that slide one more time. So this was the slide, right? So here we can see dot dot means we have much more data. So here we have these genes and we want to find out that is this gene being used more in this patient, or is this gene being used less in this patient? So we want to answer the questions like this, okay? And here if you have a lot of data, you cannot just answer that by looking at this table. So this is where R helps. So R here, it can compare thousands of genes in a few seconds and give you the result. Okay. Now, I have one more small question for you. Okay, so here I have one question for you. So just imagine a situation that you have one skin cell and you have one brain cell and they both have the same DNA. Now tell me, why do they behave differently? by your answers above, I already understand that you have a lot more information about these concepts, but we will still go through it. Okay. So the question is, if you have one skin cell and you have one brain cell and they both have the same DNA, so why do they behave differently? Okay, very good part. Okay, very good, Aryan. Okay, very nice answer, Dr. Siddhesh. Okay, very nice, Diba. So I think we all understand the answer, because in skin cell, only those genes are switched on which is related to their function, and in brain cell only those genes are turned on which is related to brain function. Okay, very good, Ragini. Okay, Saimat, because of their gene, very good. Okay, so here from now on, we will not just learn what is R. Okay, here we will learn how to think like a bioinformatician. So that means whenever we learn a programming concept, you will already understand the biology behind why we need it. So this way, R will never feel like a random code, and it will feel like a tool for solving the biological problems. So let's connect everything that we have learned so far. So we learned that every cell has same DNA, and DNA contains thousands of genes, and different cells, they use different genes, and gene expression tells us that which genes are being used, and at the end R helps us analyze that data. So can we see how everything is connected? If you just learn R concept, means if you just learn the syntax of R and you do not learn what is the biology behind it, so you will never understand what problem you are trying to solve, means what biological problem you are trying to solve. So do we understand that how everything is connected with R? Okay, so one more thing that I want to tell you something about our classes. So I have taught many students programming, and one thing that I have noticed is that many people, they learn the code that like they are memorizing the commands. So we don't have to do that. Okay, I don't want that for you, that when we will write our code, I don't want you to memorize the commands. So because what will happen after a few weeks, you will just forget everything that you learned. Even if you practice the R syntax, you will just forget that why you are doing it, how to do it, because if you do not understand the thing behind it, or the biology or the question that you are trying to solve behind it. So I don't want that for you. So instead, I want every R command to answer a biological question. So again, let's take the same example, that which gene has higher expression. So if you want to find out that which gene has a higher expression, so what you will do, you will write the programming concept related to that. So you have a question in mind that you want to solve this question, that who has the higher gene expression, or which gene has the higher gene expression. So that's where you will learn the R syntax, and that's where you will write the whole concept by yourself. So if you understand the biology behind it, so writing the prompt, or I can say the programming concept, the code, it will become much more easier. Okay, so when we finally write the R code, you will know exactly what question you are trying to answer. So here these we have 4 to 5 basic biology concepts that I want every one of you to understand clearly before we write our first R code. So they are very simple here, but they make everything very easier later. So here is the order. First thing, what is gene expression that we cover today. Second is what is gene expression data. Third is what is data set. Fourth is how does a computer see biological data. And fifth is why do we need R to work with that data. So I think we have already got some idea about this point, that why are we using R for the data. So only after these concepts we will write our first R code, okay? And by then the code will not feel random. It will feel like a natural tool to solve a problem. So this was about today's session for you to understand that why should you learn R, or I can say why you are learning R. So are we clear with today's session? Do we have any kind of questions? Okay, any question, any type of question you may ask if you have. And let me just take your attendance. Okay, just wait for 2 minutes. Let me take your attendance. Okay, thank you, Aryan. What about others? If you have any kind of question about today's session, you may ask, or you can also text in the group. If later you watch the recording and any kind of questions strike to your mind, you can text in the group, or you can also personally message me about your questions. Okay? And don't worry about the time and all, you can text me anytime. Okay, so I have taken your attendance. So if you don't have any questions, you may leave the class. Okay, thank you, Ranjita. Thank you, Anand, for joining. Thank you, everyone, for joining. See you tomorrow.